Okay, so we were talking about the relationship between the cyclic AMP and the protein kinase A. So the relationship is very very strange. If adenylyl cyclase is not going to make the cyclic AMP, then the pKa will become inactive, right? So the cyclic AMP is the molecule that is going to activate the protein kinase A. So this protein kinase A, it will be phosphorylated, and the phosphate will be transferred from ATP to serine or threonine find within the target protein, right? These are specific amino acids that are responsible for that conversion. And also the cyclic AMP, it is going to regulate the activity of pKa through detachment of its two regulatory subunits from its catalytic subunits. It is a combination of two subunits. One is the regulatory subunits and the other one is catalytic subunits. So the catalytic subunits are the main things. They are going inside the nucleus, and they are going to activate certain reactions within the cells, through gene expressions. So the pKa can catalyze the phosphorylation of various proteins in the cell. So these are the examples of the cell functions. These are regulated by the cyclic AMP. And here is the function: glycogen degradation in the muscles, and fatty acid production, and heart rate, blood pressure, water reabsorption. These are the regulated functions, and these functions are controlled by the cyclic AMP through their target tissues. The target tissues are muscles, adipose tissues, kidney, bone, and every target tissue has a different function and a different hormone involved, right? Most important hormone that is present in our body in relation with the cyclic AMP is the epinephrine. The epinephrine, it is present in the muscles, adipose tissues, cardiovascular tissues, and it is responsible for the regulation of all these functions. Okay. And then the cyclic AMP concentrations, it has different functions in different cells. Sometimes they perform the same functions, but most of the time what happens that if they are present in the skeletal muscles or the liver cells, their function will be the breakdown of the glycogen. And if they are present in the cardiac cells, they are going to strengthen the heart contraction. And if they are present in the intestinal epithelial cells, and where they are going to cause secretion of the salts and water into the lumen of the gut, right? So the lesson learned from this slide is that the cyclic AMP concentration within the cell, it has different functions in different cells. Okay, so these, most of the time these reactions, these are pre-programmed. Nature has already decided that the cyclic AMP in cardiac cells, cyclic AMP in the skeletal muscles, cyclic AMP in the liver cells, they will perform this particular function. That's it. So cyclic AMP concentrations, what happen when the cyclic AMP concentrations, when the ligands are not present, when the ligands are absent, but the cyclic AMP concentration is there within the cell? So what will happen? So cellular responses can be triggered even in the absence of a ligand by stimulating CAMP production directly. And the second method is by inhibiting the enzyme phosphodiesterase that degrades CAMP. So the level of CAMP within the cell is very crucial, right? If cyclic AMP is produced non-stop, it will cause problems. If cyclic AMP is is going to be degraded too soon, it will again cause problems. So that's why nature has given catalytic subunits and the regulatory subunits to different enzymes, so that they can regulate the concentration of cyclic AMP within the cells, right? So phosphodiesterase inhibitors, these are the methylxanthines, and these are the compounds that are present in the caffeine and in the theophylline. They are present in the coffee, tea, and soft drinks. So they often relax our muscles. basically. So whenever we take tea or coffee or whatever things like that, so what will happen, that they are going to relax our bronchial smooth muscles, and that soothes our uh, our functions, especially our bronchial muscles, and that will relieve, the feelings of relief will be felt by the human beings. So the disruption of G protein signaling causes disease in human. Yes, there are two different types of diseases that we are going to study in the following slides. So what would happen if the G protein adenylyl cyclase system could not be shut off? So what will happen? Of course, if the adenylyl cyclase it is active, it is present in the active form, so it will keep converting ATP into cyclic AMP, and when you have lots of cyclic AMP, they can continue to produce the responses through the activation of different proteins in the downstream reactions, right? So here these are two examples. The Vibrio Cholera, it is a disease by which heterotrimeric stimulatory G protein is involved. So G protein is activated indefinitely, right? So as I told you that GS and GIS stands for stimulatory, that stimulation must be regulated. Once that stimulatory protein, it is it is enough, and cyclic AMP levels remain high, and then this GS protein must be shut off. But if this cannot shut off, what will happen? The lots of salt and water will be lost in the intestinal cells, and if it is not treated, this may lead to death because of the dehydration. Okay, so here, and another example is the pertussis toxin. This is the toxin secreted by a pathogen, and on its effect on the GI protein. So GI is no longer inhibits adenylyl cyclase. As I told you that GI is to stop certain reaction. GI is going to inhibit certain reaction already going on in the cell. So what happens, that if this toxin is going to bind with the GI is no longer inhibits adenylyl cyclase. So what will happen? The resulting fluid again will be accumulated in the lungs, that will lead to whooping cough. Okay, so these signal transduction reactions, they are tightly regulated within our cells, within our bodies, tissues, organs, everywhere. And these are some of the examples. This is the last slide. As far as G protein coupled receptors are concerned, here you can see the platelet activation, muscle contraction, insulin secretion, amylase secretion, glycogen degradation, antibody production. All these have again some target tissues, right? And again they have some messengers. The primary messengers, they are going to bind on the receptors of the target tissues, and these target tissues are going to activate and regulate these functions. Okay, thank you very much.